Hello everyone, so today we are going to be discussing how bacterial transformation works. To get started, let me tell you something about bacterial transformation first. Bacterial transformation is a process of horizontal gene transfer by which some bacteria take up foreign genetic material or naked DNA from the environment. The process of gene transfer by transformation does not require a living donor cell, but only requires the presence of persistent DNA in the environment. The prerequisite for bacteria to undergo transformation is its ability to take up free extracellular genetic material. Such bacteria are referred to as competent cells. There are four main steps in bacterial transformation. The first one is preparation of competent cells. The protocols for preparing competent cells vary by whether transformation is to be achieved via heat shock or electroporation. E. coli is the most common bacterial species used in the transformation step of a cloning workflow. Since the natural competency of E. coli is very low or even non-existent, the cells need to be made competent for transformation by heat shock or by electroporation. Once prepared, competent cells should be evaluated for transformation efficiency, aliquoted to small volumes to minimize freeze, thaw cycles, and stored at an appropriate temperature to maintain viability. For constancy and to save time, pre-made competent cells are available in ready-to-use formats from commercial sources. These competent cells are quality controlled and tested to meet specifications for transformation efficiency, and genetics.
The second step is transformation. The two most popular methods of bacterial transformation are heat shock of chemically prepared competent cells and electroporation of electrocompetent cells. When ready for the transformation step, competent cells should be thawed on ice and handled gently to retain viability. Cells can be mixed by gentle shaking, tapping, or pivoting, but vortexing should be avoided. With chemical transformation, chemically competent cells are mixed with plasmid DNA and briefly exposed to an elevated temperature, a process known as heat shock. First, cells are incubated with DNA on ice for 5 to 30 minutes in a polypropylene tube. For successful chemical transformation, 50 to 100 ml of competent cells and 1 to 10 ml of DNA are recommended. When a ligation mixture is used as a transforming DNA, purification prior to chemical transformation is generally not required.
The third step is the cell recovery. This step improves cell viability and cloning efficiency. For electroporated cells, growing the cells as soon as possible is recommended, since electroporation buffers are not formulated for long-term cell survival. In the recovery step, transformed cells are cultured in 1 ml of pre-warmed SOC medium at 37 Celsius with shaking at 225 rpm for 1 hour. SOC medium, which contains glucose and MgC12, is recommended to maximize transformation efficiency. Use of SOC medium instead of Lennox L broth can increase formation of transformed colonies 2 to 3-fold. The final step is cell plating. After growing in SOC medium, the cells are plated on LB agar with appropriate antibiotics or other agents for identification and recovery of successful transformants. Before cell plating, the plates should be pre-warmed to a favorable growth temperature and be free of condensation to prevent contamination in mixed colonies. The amount of cells plated should produce a sufficient number of individual, distinct colonies for further screening. Cells cultured in SOC medium may be pelleted by centrifugation for 5 minutes at 600 to 800 xg and resuspended in a smaller volume for plating. Even distribution of the cells on the agar plate is critical for analysis of the colonies. A sterile hockey stick or L-shaped cell spreader is commonly used to spread the cell suspension while gently rotating the plate. Avoid puncturing the agar surface while spreading the cells. Alternatively, autoclaved glass beads may be used to spread the cells. After spreading, allow the plate to dry before incubating overnight at 37 Celsius in an inverted position. The culture plates are examined the next day for colony formation. Prolonged incubation should be avoided, as it often results in fusion of large colonies and the appearance of smaller, antibiotic-sensitive surrounding colonies due to antibiotic breakdown around large colonies.
That's all on bacterial transformation. Thank you for listening.